He kōna I purangi tēnei nā te reo irirangi o Aotearoa.
And so here is a trial with perennial ryegrass, our main pasture plant. Even though you can't see the fungus that lives inside the grass, it's a very important fungus, and we call it an endophyte because it lives within. Both plant and fungus live together in a symbiosis where they both receive benefits. The fungus receives a safe home and nutrients from the plant, whereas the plant, most importantly in regard to farmers' perspective, is that the fungus produces these compounds which protect the grass from the main pasture pests.
Kia ora, nau mai haramai ki te ao hurihanga. Hello and welcome to Our Changing World. Ko Clark and Canon tēnei. That's the voice of Dr. Linda Johnson explaining what an endophyte is to Veronica Meduna for a 2013 episode of Our Changing World. This week, Dr. Linda Johnson and the Endophyte Discovery team at AgResearch have won the Maine Prime Minister's Science Prize, which is awarded for a transformative scientific discovery or achievement. Over the last 4 decades, this team have been investigating some of the fungal endophytes that you find in ryegrass. As Linda said, it's the most commonly grown type of grass for livestock in New Zealand. Now, some endophytes aren't great because they can impact the livestock that eat them, but others can really help pasture productivity. The group has commercialised a few endophyte strains now, including one released in 2006 called AR37, which protects the ryegrass from a whole bunch of insects. The Endophyte Discovery team are now using gene editing techniques to create and research new endophytes, and they're investigating some that might help grain crops to grow better. You can hear more about their award-winning research on RNZ's Saturday morning show this week. Each year, a prize is also awarded to a science teacher, and this year it went to Dr. Aidan Kiley from RNZ College in Auckland. The MacDiarmid Emerging Scientist Prize went to Dr. Olivia Harrison from the University of Otago for developing a multidisciplinary approach to understanding and managing anxiety. We spoke to Olivia about her research last year in an Our Changing World episode called Anxiety and the Brain-Body Connection. It's totally fascinating research and I'd recommend a listen. In a recent update, Olivia said her group are currently finishing off their studies looking at the effects of both medication and exercise on anxiety, and she plans to spend the prize money that comes with the award on analysing their data with cutting-edge techniques. And the Prime Minister's Science Communication Prize, well, that went to someone whose voice you've also heard before on Our Changing World.
I never really set out to be a science communicator.
This is Professor Jemma Geoghegan of the University of Otago.
I felt like I had quite niche expertise in an area which suddenly became really important.
Scientists are waiting for a sample from the Air New Zealand crew member who tested positive for COVID-19 after a flight to China.
The only way we can determine where and when this person became infected is to look at the genome sequence and compare it to the genome sequences that we have already for the cases that we've had in New Zealand.
Now, the Ministry of Health has confirmed at least one case of the Omicron variant in a managed isolation facility in Christchurch or Tautahi. The variant is spreading quickly overseas and scientists are racing to find out as much as possible.
So Omicron, we're seeing overseas doubling time of about 2 to 3 days, meaning that once Omicron is in the community, the risk of it spreading far and wide are very high.
And all of this was indeed right up Gemma's alley, because her research is focused on viruses, and in particular, trying to understand how they evolve and jump to new host species.
They are the most abundant biological entity that exists on our planet. We know about probably way less than 1% of the viruses that exist on Earth. And so there is so much more to explore. Our sort of biased view of viruses and the study of virology was really from a clinical perspective. So the study of disease and how they cause disease in both humans and animals. However, the more we study them, the more we realise that most viruses don't cause disease and they seemingly exist with their host. And whether or not that interaction doesn't cause a problem to the host at all, or sometimes it could even be beneficial to the host. So there's a lot still to learn.
In 2022, I visited Jemma's lab to speak to some of the team. One researcher was investigating viruses that live in the tuatara gut, while another was trying to figure out what was causing the endangered hoiho, or yellow-eyed penguin, to get sick. Some of the penguin chicks were suffering from a type of respiratory syndrome, which resulted in many chick deaths. And the team did manage to figure out that it was a virus causing it.
What my research tries to understand is what makes a virus pathogenic or able to cause disease, and how can we recognise it when it is causing disease versus when it's just hanging out in a host. So a lot of my research tries to investigate wildlife disease, for example, where there's a disease but it's caused by an unknown, probably a pathogenic agent. And so we use advanced genomics technologies to identify what might be in a given sample from a diseased animal.
And because of this expertise in applying genomic technologies to understanding viruses, Jemma became a key part of New Zealand's research response to the COVID-19 pandemic. She worked with colleagues at ESR to sequence variants and to track the transmission of the virus.
Given that I was heavily involved in the response and doing the genome sequencing and reporting it, then it was important for people to understand what those data were telling us, not just the public, although that was really important, but for people making decisions about what we should do with this information, as well as other people, in other communities and in organisations as well. So that's kind of where it started, I guess. But the pandemic's not over by any stretch. But I guess the focus of my communication has shifted to more other things that are, in the forefront of people's minds, such as avian influenza virus or bird flu, and other disease outbreaks as well.
Do you know how many interviews you've done?
No, I stopped counting when I got to over 200, I think. So, it's funny because for the most part, I actually often decline interviews because I see the questions that they want to potentially ask me, and I see it outside of my expertise. And I feel quite strongly that I want the public to trust me in terms of I'm the right person to answer these questions. And a lot of the time it seems that a journalist might want to ask me a question that is outside of my expertise. So I do decline a lot. But having said that, during, the pandemic, I was giving interviews multiple times a day, even to the same journalist multiple times a day. So.
Yeah, you definitely became one of those voices on the radio, for sure.
Yeah.
And, you said you never had a plan to kind of go into science communication, but obviously when the pandemic hit, you kind of stood up at that time. You're like, well, I have expertise in this area, and now I'm working in this area, and I'm working at the forefront of this genome sequencing response for New Zealand. But still, you didn't have to stand up. Why did you feel that was an important thing to do?
I guess I have quite a strong sense of public duty, especially being an academic, having expertise in a certain area. I think part of the job of an academic especially in a publicly funded institution, is being critic and conscious of society, standing up when there is something that is in your wheelhouse that you have expertise to express your expert opinion about. This was a really sort of scary and uncertain time for all of us, I think. As, borders were closing, we were locked down at home. And so trying to understand more about something makes it less scary. And so that's kind of where I felt like I could help in a way.
This is not your first Prime Minister's Science Prize, right?
Correct. So in 2022, I accepted the 2021 Prime Minister's Prize for Emerging Scientists, which recognises research excellence for an emerging scientist, which I think I've emerged now. So, which was, very surprising to even have won one award, and now two is kind of just shocked, I think is the right word.
As part of the award, you get a cash prize of $55,000 to put towards a science communication activity or professional development. Do you have plans of what you're going to use the money for?
Yes, I do. So one way that I hope to use this money is to run workshops that provide communication about a genomics toolbox for pandemic preparedness and how that can be used across different interfaces, whether it's human health, animal health, in agriculture, animal health and wildlife conservation, and how genomics, because that's my area of expertise, can help and be integrated into, public health or animal health in a way that it's currently not.
Of course, part of pandemic preparedness is keeping an eye out for what might be next. One of the viruses currently spreading around the world and hitting the headlines is a type of bird flu. It's often referred to as HPAI, highly pathogenic avian influenza, or by its strain name, H5N1. Because, to be clear, not all bird flu is the same. There's bird flu found in some wild birds here in Aotearoa that mostly just hangs out and doesn't cause much trouble. Though we did have an outbreak of a pathogenic strain of bird flu at one Otago poultry farm last year. This was H7N6 type, and luckily it was quickly brought under control.
Chickens are being culled from an Otago egg farm after some birds tested positive for a strain of bird flu.
But this H5N1 highly pathogenic version that's been making its way around the world and has jumped to other hosts, not just birds, is what's concerning a lot of people, including Jemma.
The mammalian adaptation is something we've never seen before, and this is why it's becoming really problematic. So for the past 30 years, we've known about highly pathogenic avian influenza virus, but only since about 2020 or 2021 has this new lineage spilled over and is now affecting lots of different species, killing millions of birds worldwide. It spilled over to other animals like marine mammals, and at the beginning of 2024, it was found in dairy cattle in the US. And as I said before, we didn't think that this virus could spread between mammals, but it spread very rapidly between cattle farms and is now infecting sheep. It's now spread to humans as well, although there's little evidence to suggest that it can now transmit between humans. But this is something that's really concerning and quite unprecedented in the sort of what we knew about avian influenza virus.
And am I right in saying the more spillover that you have and the more kind of multiplication of this virus across the globe, you're just giving it more chances to potentially mutate into something that could spread to other mammals, other animals, and also humans more easily? I know you're saying it's already spreading to humans, but that we would start to see human-to-human transmission.
That's exactly how it works. So the more chances we give it to better adapt to those lots of different hosts, the more likely it might be to adapt to something new like humans. It's quite interesting the way it's transmitting between cows in the US because it's not causing a respiratory disease like the flu does with humans and other mammals. And so it's causing disease in udders, and it can cause conjunctivitis, things like that. So the humans that it's affecting are people that have direct contact, like farm workers, with the cattle. And for the most part, they're not getting respiratory disease. They're getting the disease that the cattle are getting because it's infecting the cells not in the respiratory tract but in other parts of the body. The reason why it's not become a human pandemic yet is it's kind of infecting the cells that aren't really promoting human-to-human transmission. For example, if it got into respiratory tract of farm animals, for example, and it adapted to mammalian airborne transmission that way, it's more likely it would become a human-to-human problem.
Right. That sounds pretty intimidating and scary.
Yeah, I mean, I think it's important to also realise that avian influenza virus, although it's not a huge problem for humans yet, it's a huge problem for wildlife. it has killed millions of birds worldwide. And, New Zealand is home to many endemic bird species that only exist here. And they're already, grappling with extinction and the threat of extinction. So I think looking beyond just human health, it would be absolutely devastating if it does arrive here because of the loss of biodiversity.
I want to ask, because you have all this knowledge, you're working in this space, you're researching virus evolution, you're communicating around pandemic threats. Is there a lot of chatter in your brain about all of these things that might happen? We've just talked about a very, potential scary arrival of avian influenza and what that might mean for New Zealand and even potential mutation, that there might be human-to-human transmission. Do you live with that knowledge just churning away in your brain?
I try not to think about it, to be honest. No, I think, so going back to, I guess, the start of our conversation is, where a lot of my research just tries to better understand this virus sphere and how there's so many viruses out there that are just seemingly existing with their hosts and not causing a problem. But only a very small amount actually cause problems and cause disease outbreaks. You kind of realize how rare disease outbreaks and epidemics and pandemics are because viruses are everywhere, but only a tiny proportion of those end up causing a disease outbreak. So that kind of is reassuring in some odd way.
In terms of viruses that go on to become pathogenic, is this just like a pure random chance numbers game?
In a lot of ways. I mean, there are multiple factors that have to align for a virus to emerge into a new host. And then there are even more things that have to align to then allow that virus to spread between that new host and spread within that population. And there's a huge element of luck or unlucky, I guess, whichever way you look at it, in terms of mutations and whether or not that mutation is particularly advantageous for the host that it's currently in. And then the alignment of the interaction with a new host species, and if that interaction is enough to allow that virus to jump those barriers to a new host species. But there's lots of barriers stopping this. I mean, viruses are quite well adapted to the host that they're really happily transmitting in, and it's quite unusual for viruses to be able to transmit to multiple different host species.
And I realised, you just went, actually, there's millions of viruses, we only own 1%. And I immediately went straight back to the pathogenic side.
Yeah.
Do you ever want to have like an interview where you just talk about, really bland viruses that are just doing their virus thing and not causing any disease? You're like, let's talk about those guys.
Yeah. I mean, for the most part, we don't really understand what they're doing there, whether or not they did cause disease when they first emerged. Was that millions of years ago? And is that what shaped that host's evolution? And the survivors managed to live in harmony with those viruses, so we don't know a lot about how viruses evolve and whether or not they always evolve to become completely avirulent or not pathogenic with their hosts. I don't think that's the case. I think it's whatever benefits that virus, right? So if the virus is benefited in some way from it being pathogenic, it might evolve that way. But yeah, there's a lot of questions.
Thanks and congratulations to the University of Otago's Professor Jemma Geoghegan, winner of this year's Prime Minister's Science Communication Prize. And last, but by no means least, is the Prime Minister's Future Scientist Prize. It's awarded for an innovative project by a secondary school student in science, maths, technology, or engineering. And this year it goes to Rena Misra from Epsom Girls Grammar School in Auckland. Just like the winners of the Big Science Prize with their ryegrass endophytes, Rena has been investigating the power of a plant-fungus combo.
My project looked at how plants inoculated with a type of fungi called arbuscular mycorrhizal fungi could enhance their ability to uptake and reduce copper contamination in water in hydroponic systems.
Rena showed that this fungus, which you can source from garden centres, helps plants suck up contaminants out of water, and therefore it could potentially be used to help clean up stormwater.
Arbuscular mycorrhizal fungi, it has a number of benefits. So one of the things that it does is it colonizes plant roots. So it sets up a network of fungal threads called hyphae. And what it does is it extends beyond the plant's root network, and it expands its surface area. So this allows it to not only absorb more nutrients to grow, so the plants grow larger and stronger, but it also means that it's able to uptake more of this contamination, so more copper from the water. And this could be really useful in reducing contamination in our urban waterways, especially because of the issue of stormwater runoff in New Zealand. So harmful pollutants, especially heavy metals like copper and zinc, after every rainfall event, it flows directly into our waterways, and it's really threatening the health of our waterways and also aquatic ecosystems. So one potential application could be putting these supercharged plants onto floating rafts and placing them on these waterways that don't have access to existing stormwater treatment infrastructure. So that could be a good, low cost and accessible way for these rural communities that don't have this technology.
Sounds neat, right? But how did Rena go about testing this? Well, it involved buckets to build her own homemade hydroponic system.
I conducted my experiment in 3 stages. So the first stage, I transplanted my plants into hydroponic systems. So half of my plants, I applied this fungi to their roots, and then I transplanted them into my hydroponic system. So I used spinach and lettuce plants, and I cultivated it in these hydroponic systems. And then after this growing period of 30 days, after that, I performed a staining procedure. So this allowed me to actually view the structures of the fungi that had colonized the roots under the microscope to see if they'd become active in my plant roots. So then, after I confirmed this, I could move on to my 3rd stage, which was I spiked my hydroponic systems with copper. After using copper test strips and RGB colorimetric analysis, I could measure how much the plants were able to uptake this copper and reduce the contamination.
Can I just jump in here to note how impressive this is? Hydroponic systems must be quite finely tuned for plants to happily grow. She had help from a mentor who runs a hydroponic chive farm, which she visited to learn how you go about it. Rena had to keep a close eye on the ion, pH and nutrient levels in the system, set up in the study room she shares with her sister.
I heard the water pumps going almost for a month, I think, yeah. And it was right next to my sister's table, so we always joke that she took care of my plants and yeah.
And because she didn't have access to lab instruments that would be able to accurately detect copper levels, she devised a workaround.
I had to think outside the box. So what I did was I took regular copper test strips from a regular plant store, and then I obviously dipped it in my systems, and then using my smartphone, I downloaded this app that picks up RGB values of the test strip simply by taking a picture. So by taking a picture of the test strip, I was able to convert the color of the test strip into discrete values of red, green and blue, so that's what RGB is. And then these discrete numbers allowed me to compare the copper levels directly rather than just relying on the color on the test strip, so it gave me numerical data.
Rena's results showed that the 2 plants that were inoculated with the fungus were bigger, had longer roots, and after 3 days had taken up more copper than her 2 control plants, which weren't inoculated with the fungus. She's enjoyed the satisfaction of problem solving and learning that if she puts her mind to something, she can do it. But the project wasn't without its challenges.
It was definitely a huge undertaking, getting all of the systems in check, set up, getting the water pumps installed, and they actually broke along the way. So I had to fully replace all of them. At the start, it was definitely difficult. I remember a few days into my experiment, I came in, all of my plants started to look yellow because turns out I didn't add enough nutrients. But then through these kind of experiences, I learned, I modified different things, and then, it turned out successful in the end.
Successful is right. She first developed the project for entry to the NIWA Science Fair, in which she won first in the senior science category and best overall exhibit. Rena is in year 13, taking 3 science subjects. In terms of what comes next, she's thinking about pursuing medical research, in which case the $50,000 prize money to support her tertiary education will come in real handy. Thanks to student Rena Misra from Epsom Girls Grammar School in Auckland, and congratulations to all the prize winners. This episode was produced by me, Claire Kincannon, with production help from Ellen Rikers and sound engineering by William Saunders. Check out the show notes for this episode or visit rnz.co.nz/OCW for more details. Tēnā koe I whakarongo mai. Thank you so much for listening. I'm Claire Kincannon. Have a great week. Kia pai, te wiki.